In 2022, a team of researchers published the results of an ancient DNA project that had analyzed genetic material from woolly rhinoceros specimens spanning the final 50,000 years of the species' existence. Bones, teeth, and frozen tissue collected from permafrost sites across Siberia, representing the most complete temporal genetic survey of any Pleistocene megafaunal population yet undertaken. The study, led by Axel Barlow and colleagues, was designed to answer a question that had been debated since the woolly rhino's extinction was first being documented. What actually killed it? The standard assumption in popular accounts, and in a significant portion of the scientific literature, had been that human hunting pressure drove the woolly rhinoceros to extinction as part of the broader Pleistocene megafauna collapse in which most of the world's large mammals disappeared between 50,000 and 10,000 years ago. The ancient DNA results did not support this assumption. The genetic data showed that woolly rhino population sizes had been stable or increasing in the millennia immediately before the extinction. Genetic diversity was well maintained. The population showed no signs of the progressive decline in effective population size that characterizes species being driven toward extinction by sustained hunting pressure. Something had killed the woolly rhino. It was gone by approximately 10,000 years ago. But that something was not a slow-motion population collapse driven by sustained human predation. It was something that happened fast, at the very end, and the genomic data pointed toward a single candidate: the rapid climate warming at the close of the last glacial maximum, which transformed the cold, dry mammoth steppe that woolly rhinos depended on into boreal forest and wetland too fast for any large mammal to adapt to through evolutionary change. The study was not the final word on the subject. The debate about the relative contributions of humans and climate to Pleistocene megafaunal extinctions is ongoing, but it was the clearest genetic evidence yet that the woolly rhino, at least, was not a species in decline when it vanished. It was a thriving population erased by a world that stopped being the world it was built for. Rhinoceroses belong to the order Perissodactyla, the odd-toed ungulates, alongside horses and tapirs. This relationship, well established by both morphological and molecular data, places rhinos in a lineage that has been distinct from the even-toed ungulates, cattle, deer, pigs, hippos, whales, since the early Eocene, approximately 55 million years ago. The Perissodactyla were far more diverse in the Eocene and Oligocene than they are today. The order once contained dozens of genera across multiple families, occupying ecological niches from small, agile browsers to the enormous Paraceratheres, the largest land mammals ever to have existed, reaching heights of over 5 meters at the shoulder and weighing up to 20 tons, larger than any dinosaur that walked on land. What survives today is a remnant of this former diversity: 3 families, 17 species total if you count all extant horses, rhinos, and tapirs. The family Rhinocerotidae itself was far more diverse in the Miocene, with genera distributed across Africa, Europe, and Asia in multiple ecological roles. The 5 living species, white rhino and black rhino of Africa, Indian rhino and Javan rhino of Asia, and the Sumatran rhino, are the last survivors of this radiation, and all 5 are currently classified as vulnerable, endangered, or critically endangered. The genomic work of the past decade has restructured our understanding of how those 5 species relate to each other, and to the extinct Pleistocene lineages, revealing that the family tree of living rhinos is considerably more complex than earlier morphological classifications suggested, and that some of the most important evolutionary distances within the family had been systematically underestimated. The most dramatic revelation from ancient DNA about rhino prehistory concerns an animal that was not thought to have much to do with the extinction crisis affecting living rhinos. Elasmotherium sibericum, the Siberian unicorn, was a giant rhinoceros of the Pleistocene, bearing a single enormous horn estimated at up to 2 meters in length on the largest specimens, that belonged to a separate rhinoceros subfamily, Elasmotheriinae, which had been diverging from the lineage producing modern rhinos for approximately 35 to 40 million years. The separation is deeper than the separation between horses and rhinos within Perissodactyla. Elasmotherium was, in every sense, a deeply alien branch of the rhinoceros family, as different from a modern white rhino as a cat is from a dog. It was also substantially larger than any living rhino, with body mass estimates ranging from 2 to 4 tons. And its horn, which, being made of keratin rather than bone, rarely fossilizes directly, but whose base leaves a distinctive oval impression on the skull, was the largest horn of any mammal in the fossil record. For most of the 20th century, Elasmotherium was assumed to have gone extinct in the Middle Pleistocene, around 200,000 years ago, well before modern humans were present in its Eurasian range. This dating made it irrelevant to discussions of human-caused megafaunal extinction. Revised radiocarbon dating of specimens from museum collections across Russia, Kazakhstan, and China, published by Adrian Lister and colleagues in 2020, substantially revised this date. The most recent confirmed Elasmotherium remains dated to approximately 39,000 years ago, placing the extinction of this animal firmly within the period of modern human presence across northern Eurasia, during the same interval when Neanderthals were being replaced and when cave lions, woolly rhinos, and mammoths were experiencing intensifying human hunting pressure. What the revised dating established is that the Siberian unicorn was not a relic of a distant geological past. It was a contemporary of Homo sapiens, alive in Eurasia during the Upper Paleolithic, during the same period when humans were producing the cave art of France and Spain. The woolly rhinoceros, Coelodonta antiquitatis, occupied a different and more central place in the Pleistocene ecosystem. Where Elasmotherium was a giant specialist, the woolly rhino was a smaller, more widespread animal, comparable in size to a modern white rhino, but covered in a dense coat of reddish-brown hair that made it suited to the cold, dry mammoth steppe environment that covered much of Europe and northern Asia during the last glacial period. Its range extended from Western Europe across northern Asia to China, and it coexisted with woolly mammoths, cave lions, cave hyenas, and the humans who painted it on cave walls in France and Spain. The woolly rhino genome, assembled from permafrost-preserved specimens of exceptional quality, revealed the molecular basis of adaptations to extreme cold that are visible in the skeleton and soft tissue, but had previously been inaccessible at the genetic level. Gene families associated with fat metabolism and fat storage showed adaptations consistent with maintaining a thick subcutaneous fat layer through long Arctic winters. Hair follicle development genes showed modifications not seen in any living rhinoceros species. Temperature-sensing receptor genes showed variants that altered thermal sensitivity in ways consistent with reduced cold aversion. The woolly rhino was not simply a rhino that happened to live in a cold place. It was a rhino that had been genomically rebuilt for cold over hundreds of thousands of generations, in ways that made it exquisitely suited to the Pleistocene world and acutely vulnerable to the world that replaced it. The climate argument for woolly rhino extinction centers on what happened to the mammoth steppe at the end of the Pleistocene. The mammoth steppe was one of the most productive grassland ecosystems in the history of the planet. Vast, cold, dry, and covered in grasses, sedges, and herbs that supported an extraordinary biomass of large herbivores. When the glacial maximum ended and temperatures began rising rapidly, beginning around 14,000 years ago and accelerating through the Younger Dryas event, the mammoth steppe contracted, and was replaced by boreal forest and wetland habitats that were poorly suited to the animals that had evolved on open grassland. For an animal as highly specialized as the woolly rhino, genomically tuned for cold, dependent on the specific plant communities of the open steppe, carrying metabolic adaptations that were liabilities rather than assets in a warmer world, this transformation was catastrophic. The Barlow study's finding that woolly rhino populations were stable or increasing right up to the final extinction, rather than showing the gradual decline that would accompany sustained hunting pressure, is consistent with a rapid environmental collapse. An animal in good health suddenly deprived of the world it was built for. Human hunting may have contributed to the final push, but the genomic data suggests the primary driver was the end of the Ice Age itself. The closest living relative of the woolly rhino is the Sumatran rhinoceros, Dicerorhinus sumatrensis, and this relationship, established definitively by the Liu et al. 2021 multi-genome study published in Cell, is one of the most striking findings to emerge from rhino genomics. The Sumatran rhino looks nothing like a woolly rhino in the way that most people picture either animal. It is the smallest living rhinoceros, standing about 1.4 meters at the shoulder and weighing between 500 and 800 kilograms, roughly the size of a large horse and a fraction of the mass of a white rhino. It has two horns, the front horn typically the larger, and it is covered in coarse, reddish-brown hair that in calves and young animals can be quite dense and long. This coat is unique among living rhinoceroses. No other living rhino species has it. That shaggy coat, which has led to the Sumatran rhino being called the hairy rhino in some historical accounts, is not a coincidence. It is a visible trace of the deep evolutionary connection between Sumatran rhinos and their woolly relatives. The Liu study showed that the Sumatran rhino lineage diverged from the other living Asian rhinos, the Indian and Javan rhinos, approximately 11 million years ago, and that the shared ancestor of Sumatran rhinos and woolly rhinos was distinct from the shared ancestor of African and other Asian rhinos even earlier. In practical terms, this means that the Sumatran rhino is not simply one of 5 similar rhino species. It is the surviving representative of the oldest independent rhino lineage among living species, and its loss would eliminate a connection to rhino evolutionary history stretching back further than the separation of Africa and South America. The Sumatran rhino's own genome tells a story almost as alarming as the woolly rhino's extinction. The 2019 whole genome study by Westbury and colleagues found that the Sumatran rhino population has been declining for hundreds of thousands of years, since long before any human impact on its habitat. The current population of fewer than 80 individuals is the remnant of a much larger Pleistocene population that contracted during the same climate fluctuations that affected other tropical Asian megafauna. The genome carries the signatures of this ancient bottleneck: elevated runs of homozygosity, reduced diversity at immune gene loci, elevated deleterious variant load in the heterozygous state. The animals surviving today are carrying a genetic legacy of long-term population decline that predates modern deforestation, poaching, and habitat fragmentation by tens of thousands of years. The recent anthropogenic pressures have simply accelerated a decline that was already written into the genome. Sumatran rhino populations are now fragmented across 3 islands: Sumatra, Borneo, and possibly peninsular Malaysia, with each fragment too small to maintain genetic diversity on its own. The genomic data shows that the Bornean and Sumatran populations have been isolated from each other long enough to represent distinct conservation units that cannot be treated as interchangeable for captive breeding purposes. The northern white rhino crisis has forced a confrontation with what extinction means in the genomic age. the last male northern white rhino, an animal named Sudan who lived in a Kenyan conservancy surrounded by armed guards, died in March 2018. The two surviving individuals, Najin and Fatu, both female, both at the Ol Pejeta Conservancy in Kenya, cannot reproduce naturally. Without intervention, the northern white rhino lineage ends with them. Genomic analysis has clarified what exactly would be lost. Some taxonomists have treated the northern white rhino, Ceradotherium simum cottoni, as merely a subspecies of the southern white rhino, Ceradotherium simum, a classification that implies the loss of the northern subspecies would be tragic but not a species-level extinction event. The genetic data complicates this view. Studies of mitochondrial and nuclear DNA from northern and southern white rhino specimens have found levels of divergence at the upper end of what is typically considered subspecies range and approaching the lower end of species-level divergence. The two populations have been geographically and reproductively isolated for a period that some molecular clock estimates place at hundreds of thousands of years. Whether they are best classified as subspecies or species is a genuinely contested question, and the answer has significant implications for conservation prioritization and legal protection status. The response to the northern white rhino crisis has produced one of the most ambitious applications of reproductive technology to conservation ever attempted. Researchers at the Leibniz Institute for Zoo and Wildlife Research in Berlin, working with the San Diego Zoo and the Kenyan Conservancy, have used eggs harvested from Najin and Fatu, combined with frozen sperm from deceased northern white rhino males, to create viable northern white rhino embryos through in vitro fertilization. As of 2024, multiple embryos have been successfully created and cryopreserved. The plan is to implant these embryos into southern white rhino surrogate mothers, animals large enough to carry a northern white rhino fetus to term and genetically similar enough for the pregnancy to proceed. The genetic material needed to maintain meaningful diversity in any resulting population is limited by the small number of founders whose biological samples were preserved. Sperm, skin biopsies, and frozen cells from deceased northern white rhino individuals held in the Frozen Zoo Biobank at San Diego and equivalent facilities. But the full genomic characterization of those samples has allowed researchers to plan fertilization combinations that maximize genetic diversity in the embryos created. 12 viable embryos had been reported as of 2024, cryopreserved and awaiting implantation. The northern white rhino genome, fully sequenced from preserved samples of multiple individuals, now serves as both the blueprint for this resurrection project and the reference against which the diversity of any population produced can be measured. It is a project that could not have been conceived, let alone executed at this level of precision without the analytical tools that whole genome sequencing has made available, and its outcome will demonstrate, one way or another, whether sequencing a genome is enough to bring back what was lost along with the last living male of a lineage. Pull all of this together, and the rhino that emerges from the genomic record is an animal whose past is richer and whose present is more precarious than its physical presence suggests. The Siberian unicorn was real and was a contemporary of our ancestors. The woolly rhino was killed not by hunters, but by the end of the world it had been built for, a victim of climate change operating on a timescale that made evolutionary adaptation impossible. The Sumatran rhino is the living representative of the oldest independent rhino lineage among surviving species, a connection to an evolutionary history stretching back tens of millions of years, and it is disappearing. The northern white rhino is two animals in a Kenyan field, carrying genomes that scientists are racing to preserve and propagate before the lineage ends. The 5 living rhino species together represent the last survivors of a family that once contained dozens of genera across every continent that supports large land mammals. The ancient DNA revolution has made the losses legible in a way that bone measurements and museum specimens never could. It has also, in the northern white rhino's case, provided the molecular tools for the only realistic path toward preventing the next extinction from being permanent. Subscribe. New discoveries about lost species posted every few days.